It wasn't a problem. My internet went down. Can you hear me, Jenna?
Yeah, we can hear you. Thanks.
Okay. Sorry for the delay here. I just need to. All right. Apologies for the delay, but can everyone see my screen now? Okay. Uh, Thanks for that. And I missed the intro, so I'm going to assume I owe Jenna thanks for a nice intro, and Ruth and everyone else at the GSA for organizing this. And I want to thank my co-author here, Fabian, for originally suggesting that we do this series. And so when me and Fabian first started talking about this, we really wanted to come up with a theme, and our main theme here was what it came down to: don't trust your sensor data, really, or at least at face value. And when you're working with sensor bias, the two real tools in your quiver that you can use are QA QC. So this is quality assurance and quality control. Quality assurance is anything you do pre-deployment to help prevent bias, and quality control is anything you do after that deployment to identify bias. So in a typical sensor deployment in a lake that we might be familiar with, quality assurance would be anything along the lines of calibration, sensor orientation when you deploy it, and then you deploy your sensor. It's out there for, say, 6 months. You bring it back in. And then quality control is being able to identify any issues that you had with the data. And then it should be, anything you learn in the quality control should go back and help inform your quality assurance protocols to help with the best quality of data from your sensor. A good example of this, this is from the Jefferson Project Sensor Network, where we're checking several pH sensors post-deployment. And you can see here a couple of these pH sensors, they failed the QC test, and so they're no longer in the time series. You can see we remove them because we're bringing them in for repair or replacement. And I think a nice analogy here to how we approach this is you have this picture here. There's a bunch of women on safari. They found this nice cheetah here. They're all focused on the cheetah, taking a picture of the cheetah. And this is, kind of the known sensor issues that we tend to concentrate on. But what me and Fabian, what we'd really like to talk about with you today are these kind of unknown sensor issues. So this is what you don't know that you don't know, and these can be, the big elephant in the background you never see coming, but can have a big effect on your sensor data. I'm going to start off with two examples from the Jefferson Project Network where I work. The first one involves the YSI XO2 sonde. This is a very common sensor sonde that's used in the field. And when we switched over to more of a high-frequency data collection with that sonde, we started noticing these downward spikes in our temperature data and other sensors, actually, but I'll just be talking about temperature here. As high as, a 0.4, almost half a degree centigrade. And we noticed when we dug into it that this was associated with the sensor wiper activation. So as you can see here in that bottom picture, this sonde has a central wiper. It's a brush that goes around and cleans the sensor faces. It's a mechanical motor, but the cycle lasts only approximately 40 seconds. But the effect you can see in that bottom plot there, we were noticing sensor bias, up to 5 minutes in time here. So we identified it as, a component failure. This was some issue in some part of the sonde. We were never able to identify what it was. But it wasn't just one sensor. It wasn't just one sonde. This happened to multiple sondes. And once we realized what it was and we started talking to some of our colleagues at other networks, we found other sensor networks were having this issue here. So it's a problem with the sonde itself. And the real issue I want to get at here is that, we didn't see this when we were taking low frequency sensor measurements. So when we were taking it, say, data every 15 minutes, as opposed to the 1 minute here, we were wiping before every sensor measurement. So there was, as far as we know, if we had this issue here, every single sensor measurement that we took was potentially biased downward by a half a degree centigrade because we were only measuring after every wipe. And so this, kind of, I give this an angry elephant here because this is something that we never knew to look for until we actually kind of, ran into this problem. But, could have been years of bad data polluting our sensor network. How we ended up fixing it, I'll just point out, is that, now when we take sensor measurements, we wipe less often. So we only wipe, say, twice a day, which we can do because we're in a relatively clean lake. But we don't collect data for 5 minutes after a wipe. So we prefer to have that data gap after a sensor wipe as opposed to having bad data, potentially bad data in our sensor network. The second example I wanted to share with you has to do with the use of fluorometers to estimate biomass in a lake. So just a kind of a quick fluorometer 101, how these things generally work. There's a light source, usually an LED. It flashes a light. You can see here that that light will hit an algal cell, as I pictured here. That algal cell will fluoresce a bit, and then there's a photodetector that will pick up that fluorescence. In high light conditions, so pretty much daytime conditions, phytoplankton will tend to dissipate excess light energy as heat, and when they do this, they emit less fluorescence. This is known as non-photochemical quenching, and tends to result in a lower bias in your fluorescence data during the day. This is a known and well-studied phenomena, so I give this a cheetah here. And there are options that people have come up with. A common one is just to avoid the bias, so only use nighttime data. Another option is to actually correct the bias. So this is data from a paper that my colleague wrote, using machine learning to correct for non-photochemical quenching and high-frequency fluorometer data. And so this is a nice cartoon we put together to kind of explain what the bias looks like. In this graphic, the top, we have the blue dashed line, which represents the actual steady chlorophyll in a water column over the course of a day. And that green dashed line is your fluorescence, your measured fluorescence. So while the blue line stays static, during the day, we've got this depression due to NPQ, but at nighttime, the green and the blue line meet up. So it's a more accurate representation at night. Now, when we were discussing this paper, one of the questions that came up is that, we know zooplankton can be attracted to light. And so we were asking, are they potentially attracted to our sensor light? You can see we've got that blue blinking light there. And if so, does it matter? And so we put together this project called FIZ, fluorometer interference by zooplankton. And, what we were thinking is that here, there's a close-up of a zooplankton Daphnia. You can see it has a clear exoskeleton. It has recently grazed, so there's a fair amount of potentially viable chlorophyll in that upper part of its digestive tract. And so the question is, could our fluorometers potentially pick up that chlorophyll in the digestive tract of zooplankton? And so to answer that first question, here on the left, that's a zoomed in picture of the very tip of our sensor here. So you can see that excitation blue light. And that's taken 5 seconds after we turned on that sensor. And in the middle, you see that kind of zooplankton disco happening here. That blue and amber light flashing is the normal behavior of the LED, and you can see the zooplankton are coming underneath that light. They're aggregating underneath there. So on the right, it's that same picture now taken 50 seconds later, and you can see there's this large aggregation, this large congregation hanging out right underneath that light. So to answer that first question, actually, yes, zooplankton can be attracted to the sensor light, the sensor LEDs that we use as part of the XO2 sonde package. The next question, does it matter? So in order to answer that question, we had to deploy several sondes off a dock here in Lake George. We had a exclusion treatment where we took a sonde and we wrapped the bottom part in zooplankton mesh. So zooplankton weren't allowed to aggregate underneath the sensor. And then we had the open treatment in blue here. And that's just a normal deployment where we have a sensor guard, and zooplankton, if they were attracted, could congregate underneath there. And I think it's pretty clear. You can see at nighttime here, when you expect the highest levels of phototaxis, that zooplankton are congregating underneath our sensors and are biasing upward, a pretty significant portion of the expected fluorescence. And so again, we get a big old angry elephant here because it's something we never thought to look for, but obviously has the ability to greatly bias our data. So to answer that second question, does it matter? Yes, in certain situations, fizz can have a large effect on sensor data. And so now we have the opposite phenomenon. Now that dashed blue line is on the bottom here, and we can see during the nighttime, we can have this pretty intense bias due to fizz. And so when we take the two together here, we end up with this kind of sinusoidal wave of bias where during the daytime, we can have depressed fluorescence values due to NPQ, and at nighttime, we can have increased bias due to fizz. So we get two elephants here because we really have this kind of 1-2 punch that's really biasing our data away from that central tendency, the blue line of what's actually available or what's in the water column. And just one last point here, I'll go back. If your plan to avoid NPQ is to avoid bias and only use nighttime data, if fizz is an issue in your system, and we don't think it's an issue in every system, but if it is an issue in your system, you could potentially be overestimating your phytoplankton in the water column. Okay, so those are the two examples I had. I thought they were kind of neat. I'm going to move on to the last part of my talk, where I get on my soapbox here and talk about the importance of space and time in your sensor deployment. Space being the location and orientation of your sensor, and time being the sampling interval used. I spent a lot of time researching harmful algal blooms, so I'm going to give you two examples from the harmful algal bloom space here. In terms of space, so cyanobacteria, when they do form a harmful algal bloom, they can form these surface gums where they're highly buoyant and they all rise to the surface. This is a nice image from a puttock paper where we have in the middle there, B, we have a frozen cross section of the surface of a harmful algal bloom, and you can see the cells there. They're all congregated right up in that upper 1 centimeter or upper 2 centimeters in the water column. And so if you're interested in monitoring for harmful algal blooms and you want to know what's happening, you really want to have your sensor right there at the very top so you can measure the magnitude of what's happening here. Very difficult, though, obviously, to get your sensor in that small, thin layer. To address this partially, one of the things we've done, so on the left, we have our Jefferson Project vertical profiler, and you can see that we have a sensor sonde that we raise and lower in the water column. One of the issues, though, is because of the length of that sensor sonde, again, this is the YSI X02, we really lose visibility to the top half meter of the water column because we can't raise the sensor sonde high enough out of the water to get that top half meter. So since we're interested in harmful algal bloom research, one of the things that we've done is in the picture on the right where I've got that red circle, you can see we've taken a second sonde and deployed that right at the surface. So this is a static sonde that hangs out at the surface about 10 centimeters below the surface of the water. And in terms of the data we're now collecting, what that looks like, we've got on the bottom plot here a heat map of chlorophyll fluorescence down to 5 meters. That's taken with the profiling sonde. In the middle, we've got the same chlorophyll fluorescence data, but now from the surface sonde. And at the top, we have wind speed. So in that bottom plot, that heat map, you can see we have a bloom right around the October 15th. And where we have that lighter color, this is actually, all of that cyanobacteria in the water column rising up out of the bottom and aggregating in that top meter where we have that really dark color. So this is a really nice image of what's happening in the water column. The middle plot here, again, you can see that same pattern right around October 15th where we have an increase in cyanobacteria. But what I want to point out is the order of magnitude we're dealing with here. So you can see in the surface, we're maxing out right around 120 RFU, that's relative fluorescence units. Where in the bottom, I saturated the axis here up to 5 RFU, but I can tell you in that plot, we're probably not seeing values greater than 20 RFU. So we're looking at an order of magnitude higher aggregation of cells that we're missing in that bottom plot, but we're catching because we have a plot right at the surface. But interestingly, I want to point out where I've got that red arrow now, we seem to have this depression in the fluorescence signal. which is odd because now I've got this other red arrow point. That's when we have the lowest wind speed here. And when we have low periods of wind, that's when we really expect to have, the highest amount of cyanobacteria congregating at the surface, because wind tends to mix those cyanobacteria cells down. And what we realized happening here is that when we had that lowest wind speed, and so if you remember back to this picture, it's really that top 1 or 2 centimeters where you can get the cells all rising into a dense layer there. So even when we had our sensor, where we thought right at the surface, at the lowest wind periods where we have the thickest cells, we're actually completely missing that. Luckily, we had a camera pointed at the water, so we were able to not quantify it, but we were able to see what was happening. But it really gets the importance of location here when you're measuring these kind of ephemeral harmful algal blooms. So moving on to the last piece here, looking at time. Again, this is now we're looking at sampling interval. And I like to use this analogy of a camera shutter speed here. So here we've got 2 images, obviously the same thing, but taking it at 2 different shutter speeds. And it gives you very different qualitative aspect of what's happening here. And so I'll take you through this kind of thought experiment here. Here, this is obviously an image taken with a bad shutter speed, or the shutter speed's too slow. You look and you can see that it's a person. You can see their hands. They're probably wearing a blue shirt and black pants, and there's a wall behind them, but you really can't tell what's happening here. And so the thought experiment is, it really gets at the importance of matching the measurement sampling interval to the relevant changes in your environment. So the shutter speed here is not matched to the motion of this person here. So when you're thinking about your sensor deployments, you want to know how fast things are changing, and then how fast you're actually measuring with your sensor. So here we see we've got a couple different shutter speeds, and that one on the top left obviously is a little more appropriate for the speed. We can now see that this is a woman. She has a ponytail. She's wearing a blue vest, a blue jacket. She got black pants and black boots. So now we're more appropriately matched here with the movement. If she was standing still, all these pictures would look the same. But because there is this movement here, it's important to match your sampling interval for talking about sensors. And so the real question becomes, what information are we losing when we have a mismatched sampling interval? And so these last few slides here, this again is data taken from a vertical profiler, where I've got that red dotted line. We did have a confirmed surface harmful algal bloom. Now, what we're looking at here is downsampled data. I should say on the y-axis, we've got depth, and obviously the x-axis, we've got time over the course of a week. This is downsampled data, so as if we were profiling only twice a day with a resolution of 1 meter. And I will point out that that was what was originally recommended to us by the VP manufacturer here. And so to go back to the analogy, there's not a lot of information we can get here. This is, if you look at it, this look like a blurry image, so to speak. And if we try to extract anything useful in terms of, quantitative analysis, here you can see where we've got that red line. I don't see any real useful information here, at least in terms of trying to figure out what's happening in the water column leading up to a harmful algal bloom. Now compare that to the actual raw data that we collected. This is the same time period, but now we're looking at data collected every 2 hours with a resolution of 10 centimeters. And this is, again, to go back to the analogy, at least a shutter speed that is more matched to what's happening. Our sampling interval is better matched to what's happening in the water column. If we then try and do a little bit of quantitative analysis on that, we can see that, yes, there is this spike in the water column associated with that surface harmful algal bloom. So we're now able to get some useful information out of this data, because we're sampling at an interval that better matches the changes we're seeing in the environment. So by using an increased sampling interval, we're really able to enhance our monitoring capabilities, and we're beginning to look at important mid-water column dynamics that lead up to a harmful algal bloom. And so just for this is the last slide I have here, just to give you kind of a preview of what we're looking at. We're trying to take this idea of sampling interval and seeing how far we can go. So now we're looking at what I like to call ultra high frequency data, so sub-second data. And is there any information we can get from really collecting data pretty much basically as fast as we can possibly collect with these sensors? So to go back to that first bloom that we looked at, now on the right side, we've got, again, downsampled data. At the top, we've got 4 Hertz data, so sub-second data. Then we go to 1 minute. There we go, 1 minute. Then below that, as if the data was taken every 5 minutes, every 10 minutes, every 15 minutes, every 30 minutes, 1 hour, and then every 6 hours in the bottom. And this gives you an idea of what kind of information we may be losing when we're sampling at slower levels or lower levels here. Certainly, information on the maximum extent of the biomass. That peak drops down as we go down to different downsampling intervals. We lose short-term ephemeral events. So even at 10 minutes, which is traditionally considered high frequency, we lost one of these spikes here, these ephemeral spikes at the surface. And we also lose the ability to characterize variance. And I won't get into it now, but we were looking at some interesting work using ultra-high frequency data to predict changes in the environment. And I'm happy to talk about that if anyone has any interest in there, but that's kind of really where we're going with this idea. And with that, again, just, thanks to everyone here. I think I'd like to hand it over to Fabian right now, and perhaps we can answer questions after the two of us give our talk.
Yes, thanks, Vince. I'll try to share my screen. I guess you can hear me. Cool.
I can hear you.
Cool. So I'll present the second part of this Limnoseries. I'm working on a project that is financed by the Swiss governmental agency, the Federal Office for the Environment, with the goal of setting up a lake temperature monitoring network in Switzerland. I will talk about 2 subjects, and one is the influence of solar radiation on temperature loggers in the water, and specifically then answer the question, how does this influence the heat fluxes that are calculated using surface water temperature? And the second point is the influence that conductivity has on stratification duration, even in freshwater lakes. I'll start with the first subject, and for a bit of context, this is an air temperature sensor from the Swiss network, Meteo Swiss, and you can see that it's shielded and it's even ventilated. If you compare this now to how we measure temperature in lakes usually, obviously not shielded and even more obviously not ventilated, at least not artificially. And I was actually wondering then when setting up this monitoring network, is it actually necessary to shield water temperature sensors from solar radiation? I found some references for river temperature, but not really anything on lakes and ponds. So I made a small experiment where I tested 3 sensors, widely used sensors, like one is the Hobo MX2203 with an accuracy of 0.2 degrees Celsius. The other one is the Venco Minilog, which is a bit more accurate, but unfortunately discontinued, but still, at least in our institute, widely used. And the third is much more expensive, but also much more accurate sensor with the RBR Solo T. And specifically, I want to draw your attention on the fact that this sensor has a steel needle in contact with the water and is not a bulky sensor as the other 2, where the temperature measurement happens inside. I have 2 test sites. One is a small, very small pond next to our institute on the left side. And the other one is Lake Lucerne, which is also right next to our institute, where I installed temperature sensors at the shore at a depth of roughly 2 meters. All the sensors were installed at exactly the same depth. Every time one of each sensor type is shielded by a PVC plate, the 3 sensors in the pond are hidden below this large plate, white plate that you can see. And I also validated the sensor after this test in a calibration bath at 6 different temperatures. And then subtracted the offset for all of the analysis that I will show. I'll start with the data that we measured with the Hobo loggers in the small pond. And you can immediately see, well, this is data from beginning of June from last year. the first 2 weeks of June, you can immediately see that during the day, the non-shielded sensor shows higher values than the shielded sensor in blue. In the second plot that will appear many times now during my talk, you see in blue the temperature difference between the shielded and the non-shielded sensor. Well, it's actually the non-shielded minus the shielded, so the values are usually positive. And in yellow, you see solar radiation measured by a nearby weather station. And you can already see here that there's a really very good correlation between solar radiation and the temperature excess that you see of the non-shielded sensor. And this excess, at least for these 2 weeks, goes up to around 0.7 degrees. During nighttime, the sensors are very close to each other with a small negative difference. The picture is pretty similar for the other, I call it bulky sensor, which is the WEMCO logger. You can also see that it's warmer during sunny days, and also the extent of the warming of the sensor is on the same order of magnitude. It's around 0.6 to 0.7 degrees. Maximum values observed in the beginning of June. It's different for the RBR logger. You see here the two curves. You can already see that by eye at least, you cannot see any differences. And if you plot the differences, they never exceed actually a 0.2 degrees Celsius, even during very sunny days. There is a certain signal, but it's very minor compared to the other two sensors. So if you look at the whole time series that I took during summer last year, you still see in blue temperature difference between non-shielded and shielded sensor, and in orange the solar radiation. You see the same picture over the whole summer, which was very sunny. The HOBO and the WEMCO sensors, the non-shielded sensor heats up to 0.7 degrees approximately, and for RBR this is much, much smaller. If we look at the temperature excess, so the difference between the two sensors, shielded and non-shielded, in a scatter plot against the shortwave radiation, we can see the same picture with the RBR heating up much, much less. The relationship is pretty linear for most points, it seems. I also want to point your attention to this area here. You can see that, well, the color is the hour of the day. And you can see that for quite dark blue dots, which are roughly between 5 and 10 a.m., and also sometimes the light greenish, we have quite low temperature excess, even for high solar radiation. And this is early morning or late evening. And this is most probably due to shielding by nearby trees and bushes. So this is not a perfect experiment, but we have some effect of the surrounding topography. I now show the results for the lake. Again, with the individual sensors for beginning of June. And here the temperature difference and the solar radiation. And this is the HOBO logger in the lake. And you see that in the lake, even though it's in the shallow region, the temperature difference is much, much less. It resembles actually the RBR sensor in the pond. And the same is true for the WEMCO logger. Again, we see a temperature difference between the shielded and the non-shielded sensor with a maximum of around 0.2 degrees, but much, much lower than before. We don't have an RBR sensor in the lake because I already thought that this would not be an issue for the RBRs. After seeing that in the pond, it worked very well. So I only show HOBO and WEMCO in the lake. And again, you see that the differences, even over the whole summer period, were below 0.2 degrees, which is also shown in the scatter plot. Then I wanted to quantify how big of a problem this actually is to have sensors, bulky sensors like the HOBO and the WEMCO that are not shielded from solar radiation, if you want to derive heat fluxes from surface temperature measurements. And you actually need the surface water temperature to calculate outgoing long-wave radiation out of a water body. You also need it for latent heat, and you need it for sensible heat. And I used the relations that are implemented in our 1D model SimStrat to compare the heat flux that is calculated with a sensor that is shielded compared to a sensor that is not shielded from solar radiation. And I did this for the HOBO sensor in the small pond. You see 4 plots here. The top over 4 days in the beginning of June, from 1st to 5th of June. The top plot is outgoing long-wave radiation. The second is sensible heat. The third is latent heat. And the fourth is the sum of the 3. And you see on the very sunny day, the 4th of June, there's a clear difference in the outgoing long-wave radiation, which is to be expected because it scales to the power of 4 to temperature. So obviously the surface temperature difference induced by solar radiation will also have a difference when you calculate long-wave radiation. It's a bit less clear for sensible and latent heat, but also during this very sunny day, there's a notable difference. And you can also see it in the balance. But it's much better visible if you just take the difference between the heat fluxes with a shielded and a non-shielded sensor. And this is what I show here. So this is the difference taken between all 3 heat fluxes that I just showed, once calculated with a sensor that is shielded and once with the sensor that is not shielded. And this is the whole time series from May to September 22. And you can see that there are maximum values of 10 to 12 watts per square meter difference on sunny days. So if you compare heat fluxes at, let's say, noon, you will have quite strong differences just induced by this temperature sensor that was heated by solar radiation. However, if you take the average over the whole period, the average overestimation by not shielding your temperature sensor, a HOBO, is just 1.5 watts per square meters. So it's actually much lower than you would think just looking at the graph. And the reason is obviously that you don't have this issue during nighttime, and you don't have this issue during cloudy days. So it depends a bit how bad this issue actually is, depending on your application, what exactly you would like to know. So the take-home messages of this part are that bulky sensors like the HOBO and the VEMCO, they heat up significantly in small water bodies or water bodies that are not exposed to wind if they're exposed to sunlight, and they heat up by 0.5 to 0.8 degrees. The RBR sensor with this needle that probably equilibrates much, much quicker with the surrounding water does not really show this issue. And the problem also does not exist in larger lakes that are continuously mixed even by weak winds. We didn't see any difference above 0.2 or 0.3 degrees. And for me, a bit the take-home message of this is that you should be aware of this issue, and that these sensors like HOBO and VEMCO and also others, you might want to consider shielding them if you are working in a small or wind-sheltered water body, depending on your application, on why you want to measure temperature. This is an example that we have in a small lake in the Swiss Alps. I have a surface buoy and a weight at the bottom, and I have 4 sensors. The lake is really shallow, and so the sunlight can even reach the 4th sensor, the one at the bottom. And to be sure, I install the PVC plate at every level of this mooring. I'll go over to the second subject that I would like to talk about. And this is something that we saw when we deployed a profiler in one of our larger lakes. And it's about the duration of summer stratification, which is obviously one of the main factors influencing deep water oxygen, which again then is one of the most important parameters for ecology, metabolic activity, and so on in lakes and ponds. The onset and breakup of summer stratification, so the duration where oxygen is not replenished in the deep water are usually computed based on thermistor strings. And usually you either take the temperature difference between the surface and the bottom, or you take the density difference between surface and bottom, only driven by temperature, to calculate when your lake will be stratified and when it's not stratified. The influence of salinity, however, is rarely considered because it's usually deemed not important in normal freshwater lakes. We have since last November a temperature profiler in Lake Egery, which is a 7.3 square kilometers, 81 meters deep lake in central Switzerland. We have a profiler buoy from FlyDog, a small company in Estonia. And we have an instrument from Hydronaut in northern Italy with temperature accuracy of 0.02 degrees, and conductivity accuracy of 3 microsiemens, and an oxygen accuracy of 0.2% saturation. As I said, this is operational since last November, and the data is available in real time on our data platform, datalakes-eawag.ch. You can just go on this website and browse the data if you're interested. There's also a lot of other data sets. Some of them are real time, and you can look at this data and even download and have a closer look at it and use it. So some of the common metrics that are used, I roughly said it before already, but what I found more specifically is that density difference between surface and bottom that is commonly used, for example, in the ISIMIP 2B protocol, so the model intercomparison project, is 0.1 kilograms per cubic meters. And actually, if you start with a temperature of 5 degrees Celsius, this represents a difference of nearly 3 degrees. So it's nearly the difference from 5 to 8 degrees. And this is the threshold that is used there and is actually widely used to differentiate between a stratified and a non-stratified water body, which in the end would mean the difference between a lake that has oxygen at the bottom or not. Other commonly used metrics are a 1 degree Celsius difference or 0.5 degrees Celsius. There's also some people using oxygen measurements at the lake bottom, which is a much more direct way, obviously, of measuring mixing depth, because once you have oxygen again in the deep water, the lake is mixed. Whereas the other measures are more like indirect ways of assessing stratification breakup. A quick look at the data we get from our profiler since last November. We had some issues in winter with the cold temperatures and the mechanics of the buoy. That's why there are some gaps. You can see the stratification building up now again in May and June. This is conductivity. The range is about 60 microsiemens in this lake. You can see that how in last autumn, the lake was clearly also density stratified by conductivity, by salinity, and that now it's slowly building up again. The picture is roughly similar for oxygen. You can see that actually this lake below 60 meters has quite low oxygen content at the end of the year, so end of 2022. And you can see the oxygen coming back into the deep water around March or April. Now, here I show surface temperature in black and bottom temperature in red at the top, both from the profiler. Same as for Vince's example, the surface temperature is actually not quite at the surface because the instrument cannot really reach the surface. But for this application, this is fine. And at the bottom, you can see the oxygen saturation varying between 0 and 84%. And now I will use different ways of calculating stratification breakup, the ones that I have shown before. So if you take the threshold of 0.1 kilos per cubic meters, you would say that the lake was actually already mixed on December 12th. You can obviously see at the bottom that there's still not too much oxygen in the deep water at this point. The next threshold is 1 degree Celsius. According to this threshold, the lake should be mixed already on January 10th. Again, we have still oxygen-free water at the bottom. The frequency of the profiles here in winter is 1 to 2 profiles a day because we don't have a lot of sunlight, and the buoy is driven by electrical energy from solar panels. Sometimes internal stages actually make that we see oxygen until the bottom at the location where we measure, but there's never continuous oxygen higher than 0. If we take the next threshold, we're already at January 18, so 0.5 degrees Celsius difference between the top and the bottom. But still, there's not oxygen has not reached the bottom. And after this, actually, the lake became even inversely stratified from around end of January on. But still, the lake was not mixed. And the time when really oxygen reached the lake bottom is actually 2 months later than predicted by this density threshold. So only on February 10th, oxygen really reached the bottom. And then it stayed for 1 week, and we were sure and thought that, well, now the lake is mixed. And to our surprise, the lake became a bit anoxic again 1 to 2 weeks later in the bottom. We discussed about this. Maybe you have an idea, but we calculated the oxygen consumption that would be necessary to cause this kind of depletion again in the end of March, and it's much too high to be realistic. So the most realistic explanations for us is that in the end, there are kind of 2 basins in this lake, although they're not very pronounced. That one basin was already mixed to the bottom and the other was not, and then water came back from the other basin. But we were really surprised to see this. And the date where oxygen really then stayed in the bottom is only on March 10th. So 3 months later than predicted by the density threshold. Now I have still surface and bottom temperature at the top, but conductivity, which is a proxy of salinity, obviously, at the bottom. And you can nicely see that during this period where the lake was actually or seemingly unstable, if you look at temperature, because the bottom temperature is higher than the surface temperature, and we are above 4 degrees Celsius. It's conductivity, salinity that upholds the stratification. And the difference during these days is just 5 microsiemens per centimeter. So it's really a tiny difference of conductivity that still made the lake stratified, and there was no oxygen, or it had not mixed if you looked at oxygen. So the take-home messages from this part are that even in freshwater lakes, salinity can significantly influence stratification duration. But most of you knew this probably already, that different estimations, or let's say thresholds of temperature and density, can lead to very different durations of summer stratification. It really depends a lot on what you choose. And ideas a bit to follow this up is that you could, instead of just choosing a threshold above which a lake is stratified and below which the lake is not stratified, it would maybe make sense to have a closer look at the dynamics of bottom and surface temperature. And at least in our example, it would probably be possible to infer mixing a stratification breakup in autumn just by looking at temperature. But obviously conductivity then confirmed that it was really salinity that stabilized the lake. And a quite obvious but sometimes a bit more expensive solution to this issue was already suggested by this paper I mentioned, Engelhardt and Kirillin, but probably also others, is install an oxygen sensor at the bottom of the lake, if possible also at the surface. And if you look at the bottom oxygen, or even better at the difference between bottom oxygen and surface oxygen, you will be sure to see when exactly the lake really mixes. So thanks for your attention. Thanks again to our financing agency. Yeah, and you can see the references below. And I guess both me and Vince are now open for questions and comments.
We have a question in the comments. They asked how do you measure latent and sensible heat fluxes?
Yeah, so as I said, these are calculated using relations that are also used in our 1D model, and they depend on wind speed, water vapor content in the atmosphere, but also air temperature and water temperature. So I used meteorological data from a weather station, and I used the water temperature that I measured with a shielded and a non-shielded sensor, and then calculated the heat fluxes. I did not measure them.
Do you have a question?
Yes, I I really enjoyed both presentations actually. I had one question for Vince. Because the processes that you showed are relatively complex. I mean, and some of them are not necessarily a wrong measurement, but they like with the surface bloom, for example, how would you communicate that to a potential data user? Like I know a lot of institutes use certain flags for this. Like, do you have like this measurement is reliable or not, or do you have very different flags? How would you try to communicate these processes?
So we follow a pretty standard format with our data. So we have a pretty robust QC review. A lot of it's automated. We run several Pythonic scripts through the data to kind of catch a lot of the big issues, and then we'll also meet as a team just to review the data. And we have a series of flags for quality. So we have just a 1, 2, 3 level of flag. 1 being like, hey, there's probably something wrong here. 2 being like, we think there's something wrong. Use with caution. And 3, like, hey, only use this if you're if you know what you're doing. And then we'll also if we know something is just drastically wrong, we'll kick it out as well. And then we also have another series of like metadata flags where we'll flag a sensor calibration, or if there was someone on site, or if there's some there's a couple different metadata flags we'll keep track of as well. So the final data set that we would release to the public in a publication, a data publication, or we also have all of our sensor data published on EDI. That all comes with a series of flags, so people know if any potential issue that we might have with the data. Actually, I take that back, that we do publish all our data in EDI, but we don't publish the flag. We just give only the best version of what we have, so people don't have to try and go through. But if there are any issues, we do have a pretty serious QC system there to flag the data.
Mohammed, do you have a question?
Yeah, thanks, Vincent, Fabian. Amazing presentations. I have a question for both of you. So Vince, I think the last part of your presentation, I think will make any limnologist an easy that is this problem with spatial and temporal deployment of sensors. But I think maybe it's economically unfeasible to improve this. So how can we make the best out of the data that we already have based on the routines that we do? Or do you have any suggestions to a little bit economically move away from these pitfalls? And for Fabian, is it possible to infer an empirical equation for the non-shielded sensors in case we are exposed to these errors with correlation to the solar radiation, since it makes in the plots you showed up to 5% error in the total heat budget. Thank you.
Fabian, do you want to go first? Then I can try and follow up.
Okay. Yes, I have actually done this before. So I have used solar radiation data to try and correct the bias induced by solar radiation on the temperature sensor. In general, as you saw, the relationship is quite linear. So I guess it's not such a bad idea. Obviously, you have to be aware then also of topography and, let's say, if it's a smaller lake, how your lake is shielded from sunlight by vegetation and so on. So your the conditions at your weather station may not, well, at least for me, the weather station was not at the same place. It was a few kilometers away. So the situations of shading effects of surrounding vegetation and so on can be different, at least if you use it on a small pond. But I guess if it's a larger water body and you have a representative weather station, yeah, I guess you could give this a try.
All right, if I can try and answer your question, Mohammed. I think my philosophy, no data is bad data, right? So like the stuff I showed, it's just a way to approach and potentially improve deployment here. If I had the luxury where we're streaming data off our platforms, we usually have good solar power, so I'm not really limited by memory since we're streaming things live, and we usually have pretty good power or line voltage. So those are the two things that usually catch you up, right? Battery power or memory when you're collecting data. So if neither of those things are a problem, and you have if you're using R or some other language where you can work with some bigger data sets just because you're collecting things more often, my suggestion is if you can work with it and it doesn't cost you anything, just try and collect more frequent data just to look at your system and say, okay, I'm not really seeing anything change, and maybe you could bring it back down. But it's always nice if you have the opportunity to look at some of these different things just to see, because you don't know what you don't know. And traditional, sensor setups, with loggers, a lot of them don't assume that you have any interest in collecting data at, say, at 4 times a second. So they're just not set up for it. But I will point out, in the paper that I mentioned that's in prep right now, we did look at different sensors here and looked at 4 Hertz data collection. And one of the things that we have a series of recommendations. Sensors that are collecting data on dissolved variables. So this is, we looked at dissolved oxygen, FDOM, temperature, conductivity. It didn't, there wasn't really any benefit in collecting data at 4 times a second. Whether you downsampled it from to every 5 minutes, every 10 minutes, it was all the same, because it's a pretty homogeneous environment when you're looking at these kind of dissolved variables. It was only when we're looking at some of these more heterogeneous variables, so chlorophyll, which I showed, or turbidity, things that are measuring particulate matter, that you might get some sort of benefit, because those are the things that are changing over these very small timescales. So yeah, it really is a balance of what your research questions are, and then what your ability of your sensor is. But I think it's just my personal recommendation is have a little fun with it and see what you see, and if you don't see anything, then go back to business as usual.
Brilliant, thank you.
Jim, do you have a question?
I do. Thanks for the presentations. Those were really interesting. Maybe it's an unfair question because neither of you talked about this, but another sensor issue can be just frequency of calibration of your sensors. And I'm wondering if you can, share any thoughts beyond what manufacturers recommend, if you've done any work on how long you can deploy like a chlorophyll sensor or a dissolved oxygen sensor without it actually affecting data quality. Because I think I know Vince, that like George, you guys have an entire fleet of sensors, but that's not probably the norm for people. And if just if you played around with how long you can deploy sensors and whether instead of swapping it out, what the effect of maybe just you know, a thorough cleaning might do to improve data quality.
I can speak to what we do with the Jefferson project, and we have looked at this exact question because there is a significant time and personnel cost. You know, I say we have a lot of sensors, so every time we go out and swap out, say, 12 sensors, each sonde has 6 sensors on it. For calibrating all of them, you know, it's multiple days of work. Calibration material, depending, you know, like turbidity standard, can be really expensive. So we did look at this as a kind of a cost benefit, and I can only speak to, you know, what we found with the YSI sensor. We also have some Turner sensors, so we do have some other sensors mixed in there. But we found in general that the optical sensors we were calibrating them too often. They were holding up quite fine over the course of a typical field deployment for our vertical profilers. So, and as long as we had, you know, pre-deployment, we put them in a standard solution, and then post-deployment, we do the same thing. So we can look at any kind of drift over time. And now we'll go, you know, 4 or 5 months with some of these optical sensors without calibrating them. The other issue is that if you calibrate every month, you often get these step functions that you then have to account for in the metadata. If you can assume that you're not drifting very much, it's actually much better not to be calibrating often, because you don't have to do these kind of corrections. But other sensors, you know, like the pH sensor, that's got a reservoir. We have chloride. Again, you know, some of these sensors that will have, you know, different technologies. They need to be calibrated more often. So say for a typical vertical profiler season, you know, every month we might go out and change the pH sensor, but everything else gets left on until then. And so it's going to depend on your sensor technology. But I would say for sure, look into calibrating only as much as you need to. And I don't know if that's a really good answer, but I think we can share some of what we learned. We've written up protocols and standard procedures for this. But you know, depending on what sensor you're using, it's definitely worth your time to investigate some of this and figure out, because you can save a lot of time and money and improve the quality of your data, not calibrating unless you really need to.
Yeah, that's really helpful. Thanks.
I cannot really comment on oxygen and chlorophyll or anything, but this is exactly what I do for temperature sensors. So for the HOBO sensors, right now I service them, or I take them out twice a year, so approximately after 6 months. And then I do an in-depth calibration in the lab using a calibration bath. So I have like 50 to 60 sensors that are continuously exchanged in like 8 or 9 small lakes, and then calibrated again, and I keep track of this. And what I see until now is that, at least for these HOBO sensors, they have an initial period of breaking in somehow. So compared to a reference sensor, they change quite a bit. But after 1 or 2 years, the change is much slower. And what is also interesting, they all go towards more negative, smaller temperatures. And I would say it varies around 0.1 degrees. So from what I have seen, if I do a calibration every 6 months, which anyways makes sense because I take the sensors out and read them out, I deploy new sensors, it's not a big deal for me. I anyways have to go there. I can limit the error, like officially it's 0.2 degrees Celsius, but I'm quite confident that actually I can measure up to 0.05 with the same sensors if I have this calibration equipment in the lab.
Thank you. We have one question from the chat, talking about how our lake analyzer has a threshold for stratification of 1 degree C, but this person their study system is a well-mixed shallow lake, and they've set their threshold to 0.1 degrees C, but they're asking for any suggestions to identify thermal stratification in a mixed and shallow lake.
Well, I would argue that the threshold does not really depend on whether your system is small or big. It's rather, it should be influenced by how accurate your sensors are. In principle, I think the reason why people choose 1 degree is because it's a nice number, and because usually you have a lot of sources of uncertainty that go into your measurements. It's not also only the sensor accuracy, it can be where exactly you put your sensors. So to be on the safe side, you say it's stratified if the difference is above 1 degree. But if you're confident in how you place your data and the accuracy of your sensors, I think it makes sense to go much lower than these 1 degrees, as I showed in my example, because 1 degree can be in larger lakes still quite far away from the real mixing. I will write the link in the chat. I'm collecting data in such small and shallow lakes in Switzerland, and I have also added a few oxygen sensors in the bottom to actually check whether or the real effect of mixing when you get oxygen in the bottom water of these lakes, and whether this corresponds with different temperature thresholds in the future. And this data is openly available there. I can put the link, but I don't think I have any different suggestions for small lakes than for bigger lakes.
Jorrit?
I had another question for Fabian. Maybe I misunderstood, but I thought that for the density threshold, you only use temperature to determine the density threshold. Okay.
Yes.
I was wondering, because I mean, you kind of need information on the ionic composition, but I was wondering if you also try to convert your conductivity measurements to salinity to include it in the density difference, and if that had a strong effect or not. Because the reason is oxygen measurements can be quite difficult and expensive, but conductivity is relatively easy often. So
Yes. So the threshold I showed is just temperature, as I said. On our data platform, we calculate salinity from conductivity and also density, just assuming a typical ionic composition with this assumption. But it can nicely explain actually these seemingly unstable situations with temperature. Yeah, and it's true, you could probably also just place a conductivity sensor at the bottom of the lake, like a coupled temperature conductivity sensor. Yeah, as long as you're not too close to the sediment. I have seen that some lakes have quite a high conductivity quite close to the sediment. So if you're maybe a few meters above that, then this could be a good idea too, to follow stratification duration.
Thank you. Are there any other questions? All right, thank you so much, Fabian and Vince. This is a great talk, and thank you everyone for coming.
Thanks for hosting, Jenna.